what I do now here at the Hutch is study the structures of proteins and other molecules that they interact with DNA and RNA, and that we try and infer things about the function of the protein, so how they might work in the context of health or disease, from their structures, their three-dimensional structures. And so one question is, how do we get information about the structures of proteins and other biomacromolecules? And the answer is almost exclusively from a technique called X-ray crystallography. In order to get these structures, the experimental structures, if you're an experimental structural biologist, you purify the protein that you're interested in, which means that you get that protein in high concentration in some kind of solution with very little else, and then you purify it and concentrate it. And what's amazing about proteins, even though these are regular globular molecules, as we'll see in a second, they will form crystals just like salt or sugar or any other kind of a solution if you increase the concentration high enough. So anyway, so that's how we get most of our protein structures experimentally, is we force the proteins to crystallize by various means, and then we shoot X-rays through the crystals, and the X-rays are diffracted. They bounce off of the crystals and form patterns, diffraction patterns, which are captured by detectors, and then by analysis of the patterns of the diffracted X-rays, we can actually figure out what the structures are. And so then that gives us lots and lots of data that people like me who are modelers, who run simulations of proteins and their interactions, that data, the experimentally determined structures, that's the basis for our simulations. And it really is it provides an amazing window into the function of these molecules. So what I do a lot on a daily basis is look at structures. So this is an example, in fact, of the one that we're going to look at in a little more detail in a second. And so what we can see is the positions of all the different atoms, and it's rendered in a certain way to show particular features that I'm most interested in at the moment. And using visualization software, we can analyze the experimental data and try and figure things out about how the protein works or how some drug might affect its function.